Welcome back to part 2 of our tour of the gram-negatives. So we're going to talk about the enterics. And so whenever you see entero or enteric, that always refers to the intestine. The majority of the ones that we're going to talk about are going to belong to the family Enterobacteriaceae. So that's just a classification division. All of these are going to be gram-negative rods. Some of them are short curved rods, like the Vibrios are going to be curved rods. All of these either live in the gastrointestinal tract as normal microbiota of humans and of animals, or they're ones that cause intestinal disease, so gastrointestinal disease. Like I said, some are normal, and those are going to be the ones that are opportunistic pathogens. And then other ones are what we call primary pathogens, meaning they're not part of a microbiome, and they can cause disease in healthy individuals. So you have normal microbiota. They're the ones that are going to cause problems when they get in the wrong place or if the host is immunocompromised. And then primary pathogens can pretty much affect everybody. Biochemically, we can separate the two. It's sort of a happy coincidence. It has nothing to do with their virulence, in that these opportunistic ones that are normally part of the microflora, they can ferment lactose. The primary pathogens don't ferment lactose, and so this is a way we can use this property in identification in the lab. So that McConkey's agar is an easy one. So there's some general things about these since they all live in the gastrointestinal tract. Many of these are going to be transmitted through either contaminated food or water. So that same fecal-oral route that we talked about when we talked about protozoans and helminths. So from the feces, they're going to exit out the feces, and then somehow they get into the mouth of the next host. So if crops are watered with feces, or just poor water hygiene, personal hygiene, or food hygiene, then these can be transmitted. So prevention, of course. So grateful I live in the United States. We have purified water. There is an FDA, and well, personal hygiene. I know, I wash my hands. So these are produce two general types of diseases. All of these will produce acute diarrhea. And remember, our definition of acute diarrhea is 3 or more loose stools in 24-hour period. So usually these things are going to be self-limiting, meaning they're going to just take their course, and then the infectious agent exits out through with the feces. So these organisms, majority of them, they're going to make enterotoxins. So what you're going to get is watery diarrhea, so without invasion. And usually this is going to occur in the small intestine. When we get dysentery, remember that's when we have blood and pus. And normally these bacteria are going to be in the large intestine. And of course, to get to blood, you have to kind of go deeper than your mucosa. And so there's going to be some sort of tissue invasion associated with those. Then when these bacteria get in the wrong place, they can cause various other infections, especially if someone's immunocompromised. And then through hospitalization, the big one's going to be through septic catheterization. So not using aseptic technique, leaving that catheter in too long. So we see a lot of these can cause urinary tract infections. In women, that's common because things are exiting out the anus, and the urethra is right here, so it's a very short trek. Also, if there's improper toileting habits, so wiping back to front instead of front to back. Sometimes, too, a patient can aspirate, so fluid from their stomach comes up and then is inhaled into the lungs, and so that leads to pneumonia. And then, of course, if any of these bacteria enter into the bloodstream, we have bacteremia and we can have sepsis. So it's just some general things about all of these. So like I said, secretory or watery diarrhea. This is going to be when the microbes are just on the surface of the mucosa. They're not going to go any deeper. They're probably going to have to have some sort of fimbriae or a pili to attach. They're going to release enterotoxins, remember enteric, so toxins that are going to affect the intestine. And the hallmark example of watery diarrhea is going to be from Vibrio cholerae, which causes cholera. And it has an AB toxin, so A is that functional part, B is the binding part, so binds to the mucosa of the small intestine. And how this toxin acts is through a second messenger. If you kind of dig back to AP1 or to AP2 with endocrine, we turn on a G protein. That G protein, that second messenger, is going to be cyclic AMP, and then that is going to turn on all of these electrolyte pumps. And so it just causes all these electrolytes to be pumped out in the lumen of the small intestine, and water follows, and you just get copious amounts of watery diarrhea. Also, enterotoxigenic Escherichia coli, so not our nice friendly ones that are living there, but enterotoxigenic, they can cause traveler's diarrhea. When someone has watery diarrhea, they won't give antibiotics. Usually they just want to do oral rehydration therapy, make sure that person has enough fluids, and replenishing their electrolytes. When we get to dysentery or bloody diarrhea, that means something else is going on. So there has to be some sort of virulence factors that allow them to penetrate, because mucosa, epithelial tissue, there's no blood vasculature there. So it's going to have to be cytotoxins. Remember, cytotoxins destroy cells, and if they destroy the cells, you can erode that gut wall, so you lose that mucosa. So a great example of doing that is going to be Shigella. If you remember Shigella, when we talked about how things enter, you have these M cells that just do a random sampling of everything you've ingested, so they get in that way. They can escape from the macrophages, and then they use those actin tails. They can just start going through cells and hide from the immune system within the mucosa. And then what happens, these epithelial cells, basically they're going to slough off, we're going to get damage, we're going to get bleeding, we can get pus formation and abscesses. So like I said, when you hear that somebody has dysentery or pus in their stools, then you know that something has gone deeper. If it goes deeper than the mucosa too, then it can also go systemic. So often those diseases are accompanied with fever as well. So Shigella, Salmonella, Escherichia coli, which has the Shiga toxin, can also do that and cause dysentery. Treatment for these, this would be more of a complicated case, so then they would do antibiotics. The thing with gram-negatives, you don't want a whole lot of gram-negative cell lysis systemically. So the antibiotic use is going to be only when it's a complicated case and the patient cannot get over it without those antibiotics. But big things too, of course, oral rehydration therapy. So yeah, it can go to the blood, into the lymph nodes, bacteremia, sepsis, shock, death. So whenever you see things like fever, headache, if it changes the blood count, lymph node enlargement, then you know that it's gone systemic. Ones that can do that, so Salmonella and Campylobacter. So it just depends on the strain, how virulent it is, and if there is penetration through that mucosa. So our first one, Vibrio cholera. Like I said, this is the calling card for watery diarrhea. And it's going to enter, once again, we have that fecal-oral route. Usually it's going to be through water. So this is a disease that you're not going to get it from animals. You're going to get it from human to human contact. It is endemic in Asia and in Latin America. And you typically hear about cholera outbreaks now. There's a whole bunch of them throughout the course of history. Basically, Europe always had a lot of cholera outbreaks. But right now it's endemic in Asia and Latin America. And then what happens when there is war or a natural disaster, or you have refugee camps set up, then it tends to rip through those countries. What it's going to cause is just watery diarrhea. Okay, look at these pictures. This looks so incredibly miserable to me. The big thing they always say associated with cholera is rice water stools, meaning it's just so watery. There is nothing left, that they make cholera beds. The patient is so weak, it is so intense that they just lay there. Yes, the feces just enters out into a bucket. And what the healthcare worker will do is just to really try to replenish those fluids continually, and definitely make sure that there's electrolytes as well. Because what can happen if it's not treated, you get severe dehydration, so low blood volume, low blood pressure, hypovolemic shock, and death if they're not replaced. Thing is, there are many asymptomatic carriers. I don't know if you remember when Haiti was hit by the hurricanes, a bunch of relief workers came, some of them from Nepal, and they were carriers for cholera. And so then they spread cholera to all of the people trying to recover from hurricane. Also with these diseases, we kind of want to pay attention to infectious dose, so I put that in your notes. Thankfully, Vibrio cholera has a high infectious dose, but if the water supply gets contaminated, then that's how it spreads. Other things as far as clinically and for identification is motility. So Vibrio cholera is a cute little monotrichous polar flagella, so they are motile. I'm going to point out the ones that aren't motile. Might help you out in lab. So that is for you. Our next one is Shigella, and notice that I did not put a species on here. There are 4 species of Shigella that cause disease. Shigella dysentery, and that's kind of telling you what it's doing. Yes, it causes dysentery. Shigella flexneri, Shigella boydii, and Shigella thoniae. The big famous thing about Shigella is that it has a very low infectious dose. So just 10. Some books even say less than 10 is enough to contract the disease. And so the disease is called shigellosis. I think I just put dysentery in your notes. A distinguishing feature from the other enterics is that Shigella is non-motile. Like I said, they're going to move because they hijack the host, so through those actin tails. And once again, so we get that sloughing, we're going to get blood, we'll also get fever. You do also get nausea and vomiting. So these different strains, what's different about them, and as far as the intensity of the disease they cause, some of them have extra virulence because of plasmid genes that they have. There is another type of Shigella that has a Shiga toxin. This Shiga toxin is a cytotoxin, and it causes cell death. It shuts down protein synthesis. And so you get hemolytic uremic syndrome, which is going to lead to kidney failure. So heme for blood, lytic, lyse, uremia in the urine, and syndrome. And so you end up with bloody urine, low platelets, so thrombocytopenia, and then clotting occurs inside the kidney, so it causes kidney damage. We'll talk about this because the E. coli that you hear about in the news has a Shiga toxin gene that it got from transduction from a phage infection. So you'll hear more about that later, but I put it in your notes. So this is for you. Since there is that low infectious dose, remember all of these are going to be through, you can do it through contaminated food, water, through the fecal-oral route. But if someone has poor hygiene, you just need 10 or less, and just think about where it's exiting. So yeah. So it can also be transferred by humans specifically, but direct contact. So Salmonella, there's different types of Salmonella, and I could talk about it, but this does a nice job with pictures. So there's typhoidal Salmonella and non-typhoidal Salmonella. So let's just go ahead and watch it, and I'll fill in the gaps or point out what I want you to know.
Salmonella is a group of bacteria that can infect animals and humans. The bacteria was first identified in the 1800s and named after an American scientist called Salmon. Salmonella belongs to a group of bacteria called gram-negative bacteria. They're shaped like rods and can have flagella to help them move around. There are over 2,500 different types of the bacteria. Most human disease is caused by the bacteria belonging to a subspecies called Salmonella enterica. It's estimated that tens of millions of human cases occur worldwide every year, causing over 100,000 deaths. Salmonella can live quite happily in the intestines of many animals like cows, turtles, pigs, cats, and dogs, and in birds like chickens and ducks. Other parts of the animals, like feathers or fur, or the places they live in, can be contaminated by the bacteria. Some types of Salmonella, like Salmonella typhi, live only in humans. From these sources, the bacteria can spread to humans either directly or indirectly. For example, it can spread to people directly when they eat contaminated meat or animal products like eggs that are not cooked properly. Eggs, in fact, are a very common cause of Salmonella outbreaks. They can also spread through direct contact with infected animals or their environments. Salmonella can also spread indirectly by contaminating water, other foods, utensils like knives and cutting boards, or the hands of someone who handles food. Salmonella is one of the most common causes of foodborne diseases in the world. People with Salmonella infection can also spread the infection to others. Sometimes people can have Salmonella for a long time without showing any symptoms. So what does Salmonella cause? It can cause 2 broad types of disease depending on the type of Salmonella that causes the infection. These are typhoidal and non-typhoidal salmonellosis. Let's have a closer look. Typhoid fever and paratyphoid fever is caused by either Salmonella typhi or Salmonella paratyphi A, B, or C. These two illnesses are similar. When these bacteria are ingested, they can enter the bloodstream, leading to symptoms like high fever, stomach aches, headaches, loss of appetite, or a rash. These symptoms usually occur from between 8 to 14 days after being exposed to the bacteria in typhoid fever and 1 to 10 days after being exposed for paratyphoid fever. In some cases, it could lead to serious complications like bleeding from the intestinal tract or infection of the brain. The elderly and those with weakened immune systems are at a higher risk of complications. Non-typhoidal salmonellosis is typically characterized by a gastroenteritis and is commonly caused by salmonella types like Enteritidas, Newport, and Typhimurium. Symptoms are usually a fever, abdominal pain, diarrhea, nausea, and sometimes vomiting. These symptoms usually occur between 6 to 72 hours after being exposed to the bacteria and can last for about 2 to 7 days. Although the disease can be relatively mild in most people, it can be quite severe in the young and elderly and those with poor immune systems. Sometimes the bacteria that cause non-typhoidal salmonella can enter the blood and infect other organs like the brain or the urinary tract and cause local complications. So, how can we prevent salmonella infection? Because salmonella is mainly a foodborne disease, one of the most important ways of preventing salmonella is ensuring the safety of food. There are a number of ways this can be achieved, including making sure food preparation areas are clean, separating raw and cooked food to prevent cross-contamination, cooking food thoroughly, keeping food at the correct temperature, and using safe water and raw materials to prepare food. To reduce the chance of being infected, high-risk foods that may contain the bacteria should be avoided. This includes raw eggs, unpasteurized milk, and raw meats. Fruits and vegetables should be washed carefully, especially if they are eaten raw. Avoid using water that has not been boiled or treated. It's very important to wash hands, especially before handling food, during food preparation, after using the toilet, and after contact with animals or those who are sick. People with salmonella infection can spread the infection as long as the bacteria are present in their feces. They should not prepare food for others if they have symptoms and seek advice from their healthcare provider on when they can go back to work, school, or childcare. This is especially important for food handlers and those who care for vulnerable people. Some people, like those with typhoid fevers, may require negative stool samples and clearance from a doctor to say they are no longer infectious. There is vaccine that can protect specifically against typhoid fever. Salmonella infections are usually diagnosed by identifying the bacteria in stool samples or blood. Other types of blood tests, like those that check for antibodies, are sometimes used but are not as good. In some instances, salmonella can be isolated from sites like brain or urine if these sites have been infected. Uncomplicated salmonella infections are usually managed by rest and adequate hydration. Antibiotics are used for some high-risk groups, complicated cases, and also to treat salmonella infections like typhoidal fevers. There are some salmonella strains that are resistant to some antibacterial medication. This is a serious public health problem because these infections are difficult to treat. For more information about salmonella infections, talk to your healthcare provider and have a look at the websites below.
Okay, let's unpack that a little bit. So the genus is Salmonella, the species is Enterica, but then there's all these serotypes. So Enteritis is the one that causes just basic gastroenteritis. And so it makes it endotoxin, it has adhesins, and then of course we don't like the endotoxin. It is in other foods, and this is normally when you hear about food outbreaks, but eggs are a really big cause usually, so uncooked egg products. There was an outbreak with cookie dough, so if you like to eat cookie dough, there's uncooked eggs in there, and so there's been outbreaks with that, with chicken. Also, there was an outbreak from a pet store because of the turtles, and I don't know if you've ever handled a turtle, but you should always wash your hands after handling turtles or other reptiles because they have salmonella. So what's happening in the poultry industry, because they knew salmonella is like number 2 as far as our bacterial gastrointestinal diseases, they decided to treat the poultry with antibiotics because about a decade ago, 1 in 3 chickens just had salmonella. So in their meat, like when they handle the meat for processing and also just from the eggs, you get salmonella. And so they started treating them. So now only 10% of the chickens have salmonella, which is great, but now we have antibiotic resistant salmonella. So that's the downside. Usually, like I said, it's going to be self-limiting. Someone's just going to have, diarrhea. They may have some pus. It can spread and cause other infections, particularly if someone is immunocompromised. If someone has sickle cell disease, it can go to the bone marrow, and that causes osteomyelitis. Thankfully, it has a high infectious dose. Let's see if I missed anything. Basically just proper food handling and not eating raw foods. Like I said, particularly associated with poultry. But that's not it. I think they had a peanut butter outbreak as well. All right, so the other type. So this is the more serious or deadly type. So Salmonella enterica, serotype, typhi, or paratyphi. So I just put it in your notes as Salmonella typhi, paratyphi, and the other one I just put Salmonella enterica, enteritis. And so we're going to classify this as typhoidal fevers or typhoidal salmonellosis. This is going to be rare in the United States, but it's high in developing countries. It can kind of do everything the other one can do, except it multiplies in white blood cells, and then it can get carried around. Also, that carrier state, remember Mary Mallon at the very beginning, so typhoid Mary. So this can persist. It is a little more invasive, so it causes that rupture, internal bleeding. Even these Peyer's patches, AP2 people, those Peyer's patches, those are mucosa-associated lymphoid tissue. They'll even rupture. So you just get this internal bleeding, shock, and death. It's a little more serious. Humans are the only source of infection for typhoid and paratyphoid fevers. So you do not get this from eating infected foods. Campylobacter jejuni. This is our #1 cause of acute diarrhea in the United States. It has a low infectious dose, and so it can be transmitted person to person, but not that often. So it has an infectious dose of around like 500. You can get through ingestion of undercooked poultry or meat. It is in all kinds of food animals, so cows, pigs, chickens. Also, though, yes, our sweet pets. So from handling dogs, dog feces, cat feces as well. Like I said, it's the leading cause of acute bacterial diarrhea. So it invades mucosal surfaces. It has some toxins. Endotoxin, which is just that gram-negative cell wall, it does make enterotoxin, and it also makes a cytotoxin. So that's why you can get the bloody diarrhea. And it's systemic, so fever, cramps, vomiting. Another thing that is clinically very relevant is that after Campylobacter infection, some people develop Guillain-Barré, which is a nervous system disorder, and it causes ascending paralysis, so distal to proximal, and it is the leading cause of acute paralysis now that polio has been eradicated. And so in about 20 to 40% of the Guillain-Barré patients, they had a Campylobacter infection previously. Otherwise, it's kind of, this is one of those mysterious illnesses. A lot of times they don't know exactly what's causing it. They just know it's cross-reactivity either to a viral or bacterial infection. Like I said, a lot of the times it's going to be a Campylobacter infection. That is unique. This one, since it does have a low infectious dose, so a drop of chicken blood or a drop of meat blood would be enough to transmit Campylobacter. So yeah, that's another reason. So from Salmonella and Campylobacter, so Campylobacter is #1, Salmonella is #2. Wash those cutting boards. And that table's for you. Last one, Helicobacter pylori is not a member of the family Enterobacteriaceae. This one is going to affect the stomach. We talked about it before, so I'm just going to throw it in there because it's technically gram-negative. While it is classified as a rod, remember it has that, it's called Helicobacter. Because of the flagella, it is going to burrow into the intestinal mucosa. It can survive the stomach acidity because it makes urease and it drops that pH. So interesting story is that they used to think that ulcers, so peptic ulcer is a stomach ulcer, and so you get bleeding from that mucosa because that acid then will destroy the wall of the stomach. They used to just think people got ulcers because they were high-strung, they ate spicy foods, or they drank too much. And this particular bacterium wasn't even discovered until, I think, 1979, because they were looking at biopsies from ulcers. And these two men, Robin Warren and Barry Marshall, they were working on this project and they said, oh, these bacteria, I wonder if they're the ones causing it. At the time, people just thought nothing can live in pH 2, which is the pH of the stomach. So they actually drank, or they took an inoculum of Helicobacter pylori, developed ulcers, and therefore proving that ulcers, many of them are going to be associated with a Helicobacter infection. They won the Nobel Prize in 2005. So they made themselves guinea pigs. Also, Helicobacter pylori is associated with stomach cancers. So you have an infection, high risk of a peptic ulcer, and then peptic ulcers, you have a higher risk of developing stomach cancer. So most infections are asymptomatic, and they're just assuming that it's probably through fecal-oral transmission that people get Helicobacter pylori. They're saying incidence increases with age because, of course, our immune system goes down, but also as people age, they have a higher likelihood of developing a peptic ulcer. I'm going to stop here. Come back for part 3. You guys have a great day.